In this video, we will be demonstrating the setup for your water relations lab, where you'll be learning how different environmental factors affect the rate of transpiration in plants. So to begin our setup, we are creating what's known as a petometer, which is what we will actually use to measure the rate of water uptake in our plant. So to do this, we are adding water to the funnel to fill up the tube on the bottom left side of this image. And then once that fills with water, we are going to close the clamp below the funnel and finish filling the funnel with water. And we also really want to do our best to eliminate as many air bubbles in our system as possible. So now that we have our apparatus filled with water, we are going to take a cutting of geranium and recut a couple centimeters off of the stem underwater. Then we will insert the stem into the rubber tubing without letting any more air in. Next, we are opening up the valve below the funnel to allow water to fill up our glass pipette. And we want to be able to see the meniscus in the pipette as well, because we will be able to measure the rate of transpiration by timing how fast this meniscus moves down the glass tube. So once that's filled with water, we will close the valve again. So to start collecting data, we will first let our system stabilize for a few minutes, and then we will record the position of the meniscus under ambient conditions every minute for 7 to 10 minutes until the rate becomes stable. And these data will help establish a baseline for our next experimental treatments. Now, if we were doing this together in class, each group would be assigned a different experimental treatment for their petometer, and each treatment is designed to imitate a different environmental condition that we'll explain. So each experimental trial will run for about 7 to 10 minutes to make sure that we are able to record a stable rate for each treatment group. And the first condition we will be testing is bright light. And we can create our own bright light using a desk lamp here that is placed near our geranium. So the next condition that we will be testing is wind. And we can do this by using just a regular desk fan and have it blow air on our plant in the petometer. The third treatment we will do is increase the temperature around our plant, and we will do this by using a space heater. And then finally, for our last trial, we will be testing high humidity, and we can create a humid environment by placing a plastic bag over our plant. So now that you hopefully understand the setup of this experiment, you can begin analyzing the data to understand how each of these environmental conditions affects the rates of transpiration in plants.